This presentation on polyRAD is sponsored by the Tools for Genomics-Assisted Breeding and Polyploids Training Workshop and funded by NIFA, the National Institute of Food and Agriculture within the USDA Specialty Crop Research Initiative, and was held on January 13, 2021.
All right, so thanks for the opportunity to talk today. So I'll be discussing my R package polyRAD, which does Bayesian genotype calling from sequence reads. All right, so first, before I forget, I'm going to acknowledge the funding. So the polyRAD, the project to create this software was sponsored by the US National Science Foundation. And there are several students on this project, and Joyce Njuguna is here today to help me field questions. So if you see her in the chat, she knows a lot about this software. And for the demonstration data set, I'm going to be using some Miscanthus sacroflorus data, which was a large international collaborative effort led up by Eric Sachs via the US Department of Energy. And I'm also going to give a quick plug for the unit that I work for at the university. So I actually do bioinformatics consulting. So our unit HPC Bio, we do bioinformatics consulting, fee-for-service data analysis trainings. So if you need some help with analysis, particularly analysis of sequence data, and you're willing to pay money for it, that's what we do. All right. So when and why would you use polyRAD? So as Marcelo was kind of talking about, you have to use certain pipelines to output those sequence read depths. So in a VCF file, that would be in the AD field. And you can use options other than VCF, but ultimately what you need is sort of laid out in this table here where I have SNPs by samples. So for each marker and sample, I need to know the number of reads for the reference allele and the number of reads for the alternative allele. And polyRAD also supports multi-allelic loci. So here I'm just showing biallelic SNPs, but you could have sort of more of a haplotype-based locus with multiple alleles. But you need to know the read depth of each allele in each sample. Now, if you are starting with FastQ, there's an option installed with polyRAD. It's actually a pair of Python scripts that can do the variant calling for you as well. And those are kind of optimized for an allopolyploid genome or otherwise a highly duplicated genome, although you can use them for a non-duplicated genome just if you want to be really good at filtering out paralogous sequences. I won't discuss that today, but I just want to mention that's there. So in your data set, in this case, you have to know what the ploidy is. And you need to have hundreds of individuals of the same ploidy because the genotyping is going to be done across the whole data set at once. So the software is really optimized to work well for diversity panels and natural populations. There's a pipeline in there for biparental mapping populations, which allows any number of generations of selfing or backcrossing or intermating. I will say the mapping population pipeline is not my proudest accomplishment. I think Supermassa and Updog might be a little bit better for mapping populations, but you can always give polyRAD a try, give Supermassa a try and see how they work for your population. But if you're doing like a GWAS panel or something, polyRAD is really good for that. So output is it outputs posterior probabilities of genotypes, which is basically saying, okay, what is the probability that this genotype is the correct genotype at this marker and sample? So you can then further process those to get a discrete set of genotypes, say just taking the most probable one and calling that as the real genotype, or you can get something that's a continuous variable called a posterior mean genotype that I'll discuss later in the talk. So after the workshop, if you want more help or more discussion about polyRAD, the Slack channel I'll be watching, but also another really good option is the GitHub page. So you just have to make an account with GitHub, which is pretty free and pretty quick. So there is a discussion tab on the polyRAD site for GitHub. So that's kind of like a forum-like discussion. So that's a great way to ask questions. Traditionally on GitHub, the issues tab has also been used for discussions and questions, and that's also fine to post something there if you prefer. Issues is maybe a little bit better if you think you found a bug with the software that needs to be fixed. Okay, so what is Bayesian genotype calling? So basically there are these three components and two of them are used to calculate the third one. So the likelihood is basically the likelihood of seeing those read depths for your alleles if a given genotype were the true genotype. So the likelihood is like saying, okay, if the genotype were AAAB, what is the probability of seeing 7 reads of A and 4 reads of B? And I'm asking that because those are the number of reads that I saw. So that's what the likelihood is. The prior probability of the genotype is saying, okay, assuming I didn't know anything about the reads that I'm seeing right here, how frequently do I expect to find AAAB in the population? And then you can put those together to calculate the posterior probability of the genotype, which is saying, okay, I observed 7 reads of A and 4 reads of B. What is the probability that AAAB is the true genotype? So what are the implications of using this type of genotype calling? So if your read depth is high, those posterior probabilities are mostly influenced by the likelihood. So in this diagram on the right here, on the x-axis, those are the priors, like the width of the rectangle is showing the prior probability, and the height of the rectangle is showing the likelihood, and then the area of the rectangle is the posterior probability. So if you have high read depth, one genotype is going to be much more likely than the others. And here I'm just showing 3 genotypes for a diploid model. If I had a tetraploid model, there would be 5 genotypes. Right, so say I got like 30 reads of allele B and no reads of allele A. It looks pretty likely that BB is the correct genotype. And so the priors don't matter so much because one of them is so much more likely than the other. As the read depth gets lower, it becomes a lot harder to be certain of the genotype from the sequence reads alone. And that's when the priors start to influence the posterior probability more. One implication of this is that it helps prevent heterozygote undercalling. So say I only had 2 reads of B and no reads of A at all, but B was a really rare allele in the population. There's a possibility that the true genotype was AB, but I just didn't happen to sample any reads of A. So even though the most likely genotype is BB, because the prior is so much higher for AB, then I end up calling the genotype as AB, saying probably what happened is I missed an A allele because B is very rare. I'm not expecting too many BB homozygotes. Another implication is that if you have 0 read depth, the posterior probabilities are the prior probabilities. So this sort of becomes a form of imputation. And assuming your priors are sufficiently informative, you can use this for imputation. And the nice thing is that it's sort of a continuous scale depending on how much read depth you have between hard genotyping and genotype imputation. It's not one or the other. It's really both at the same time. Okay, so where do I get these priors? So there are a few options that come with polyRAD. There's the iterate HWE pipeline that assumes your whole population is in Hardy-Weinberg equilibrium. You can also add a selfing rate that would be the same selfing rate across the whole population. So self-fertilization. And it does that from allele frequencies to get estimated genotype frequencies. Another option is the iterate pop structure pipeline, which assumes there's population structure and it runs a principal components analysis on the data and then estimates a local allele frequency for each individual. So saying it comes from a particular part of a population, it has a specific allele frequency. And then you get a different prior basically for each individual. So then that can be good for imputation. And both of these also have an option to use linkage disequilibrium at nearby loci as well, depending on how much LD there is in your species and how densely packed your loci are. If you want to do it for a biparental mapping population, there's the pipeline mapping to parents option. Now other software have other priors. I think Marcelo talked a little bit about priors in Supermassa. And I can tell you UpDog has some priors that are based more on statistics and less on biology than these are, but they also work very well. So I mentioned posterior mean genotypes. I'll explain a little bit more of what those are. So they're good if you care about additive genetic variants for something, and you don't have high enough depth to be really confident in your genotypes. So on the left column here, I have allele copy number. So say 0 was AAAA, and 4 was BBBB, and 1 was AAAB. So these are your 5 possible genotypes in a tetraploid. And say I've done my genotyping and I have my posterior probability of each of these. What I do is I multiply that allele copy number by the probability, and I sum all of those products together. And so in this case, I get a genotype call of 1.49, which reflects the fact that it could be 1 or it could be 2, or there's a slight probability that it's an even higher copy number than that. And so then I have this sort of continuum of genotypes rather than classifying all of them discretely. And then that can be good for a linear regression with a phenotype. And on average, this reduces genotyping error as compared to just selecting the most probable genotype. All right, so the data set for today is a diversity panel with 270 tetraploid Miscanthus sacriflorus. So this is a wild grass that's being developed as a bioenergy crop. So this was collected across East Asia, and RAD-seq was performed with PST1, MSP1, and single-end reads. So what I did to call variants was I ran the tassel pipeline and exported that to VCF. All right, and so we're going to import that VCF into polyRAD. So a VCF, if you're familiar with that format, it contains genotype calls, but tassel only does diploid genotype calls. So we're going to completely ignore the genotype calls from tassel, but we're going to use that sequence read depth that comes from the VCF. We're going to do some filtering on accessions and filtering on markers and a couple of QC steps on the data. We're going to use the population structure model to perform genotype calling. And we're going to generate a posterior mean genotype matrix, which could be used for further statistical methods, like if you wanted to do genomic prediction with BGLR. And I'll also show you how to export the discrete genotypes to VCF. Okay, let's see. I think I have to stop share for a minute to switch screens here. There we go. Okay, so here we are in RStudio. So there are 3 files that you should have, although one of them is optional. I'll explain that. So you should have this workflow.r, which contains the script. The data set is this MSA tetraploids filtered data set. Now, depending on where you went looking for the files, you may or may not have gotten this FA file. If you don't have this reference genome, if you're watching this live, don't go get it now because you won't have time. It's 2 gigabytes. If you're watching a recording, go get the reference genome. It's the Miscanthus sinensis reference from Phytosome. So if you're watching this on YouTube, you can go back and get that. I'll show how to do this pipeline either way. And so you want these 2 or 3 files all to be in the same directory. If you're familiar with RStudio, I suggest setting this up as a project in RStudio. Another alternative is once you've opened this workflow.r, just go to session and set working directory to source file location. And then R will know where to find those files. So I'm going to run the library commands to load these 3 packages. So variant annotation will be used for reading and writing VCF files. And that's bioconductor, so it's loading all sorts of other packages, and it might take a minute. There we go. Obviously, polyRAD will be using for genotype calling. And QQman we'll be using to make some QQ plots for part of our QC. All right, so the first thing we have to do with this VCF is actually compress it and index it. And this will make it quick to basically quick to access specific parts of the genome if you didn't want to read the whole file at once. So I run this bgzip command. Right. And then I'll run this index tabix command. And so when it does that, it generates these two new files. And if you have downloaded the reference genome, you should also build an index for the reference genome, and that will allow polyRAD to quickly retrieve sequences from that reference genome. And it's going to need to do that because when it imports the VCF, it's going to take the SNPs and reconstruct them back into haplotypes. And so it needs those nucleotides in between to pad it out. It will work without that, but then that limits your export options after. Yeah, and I don't have the chat open on my window, so I'm going to rely on Joyce to get my attention if there's something that needs to be answered. All right, so this command below here is going to be the one for importing the data. And yeah, while we're waiting, I'll tell you right now, if you do not have the reference genome downloaded, just put a comment on this line. If you do have the reference genome, then don't put a comment on that line and leave it as it is. All right, so for me, that indexing step has finished. And so I'm going to start doing the data import. And this takes a few minutes. So not only is it reading the VCF, but it's going to, you'll see the message pop up that it's phasing SNPs. And so this is not a particularly fancy phasing algorithm like we're going to see tomorrow. This is just saying, do these SNPs appear to have originated from the same RAD tag, just based on their proximity to each other and the sequence read depth of them. If they have the same read depth, then they might have come from the same tag. And then it's going to assemble those SNPs into tag-based haplotypes, but it's not going to try to make haplotypes over longer distances. Right, but this is useful for things like imputation. There's not a lot of point in, if you're doing a GBS or RAD approach, there's not a lot of point in using SNPs within the same tag for imputation because you have the same reads for both.
So Lindsay, question that's come up is there any way of verifying ploidy from sequencing data? Or is there any way of telling the difference between an auto and allo polyploid if you do not know the ancestry?
Right, so so if you know it's auto polyploid, actually I'll be showing something in a minute that can help identify. It's not really good for identifying the ploidy itself, but it's good if there are outliers, they might be a different ploidy. That will be the H(n/He) statistic that I'm going to show next. I know someone else wrote a software for determining the ploidy from sequencing data, and I don't remember the name of it off the top of my head. The things that we're going to do here are going to be more along the lines of, like, I assume it's tetraploid. Is it behaving like a tetraploid? Or is it behaving like something else? And if it's behaving like something else, maybe I just want to throw it away. Yeah, I think the import step is based on doing this before, it's roughly a third of the way done. So I have time to answer some other questions if there are any.
Question coming up is, are the ref/alt calls based on the reference FASTA or major/minor?
They would be based on what is in the VCF and how it's listed as ref or alt in the VCF. Actually, I think in the polyRAD object itself, it no longer really defines, because it assembles them into haplotypes, it no longer defines one as the reference or alternative haplotype anymore. If you export, say, to like a matrix of posterior mean genotypes, by default, it will just omit the most common one, kind of using that as a reference. I believe when we export back to VCF, it will look at the reference genome again in order to figure out which alleles are really reference alleles.
Lindsay, could you comment about what SNP calling methods you can use prior to this, like how, where you can inherit your VCFs from? There's a question about that.
Okay, yeah. So I like to use Tassel GBS2 because it's fairly quick, but the ones that were mentioned in the last presentation, like Freebase or GATK or Tassel for Poly, all work. There's also import options in polyRAD if you use stacks, which is a little bit more popular in the like ecology sphere of research. There is an option for the unique pipeline to import from there, but the unique pipeline itself is kind of deprecated. And it won't report read depths higher than 127, which is problematic. Yeah, I know I have a couple other assorted import options, some of them for Python scripts that I have written for getting read depth, and then I have, R functions to import them from here. So that's, not other software so much. I would bring up the help page, except that R is running, so I don't have access to the help page while something else is running.
One more question, since we still have time. Is there any benefit in using Tassel over stacks for processing GBS data for polyploids?
Not that I know of. I mean, I think that they probably do different things that may or may not be documented in terms of like, how are they deciding which reads to throw away or that sort of thing. So you probably wouldn't get identical results. I mean, my main reason for Tassel is that I can run it on a laptop instead of, whereas stacks, you would really need like a high performance computing environment. And the reason why you can do Tassel on a laptop is that it kind of collapses everything first into the all of the unique sequence reads for each sample, and it completely ignores the quality scores. So, I mean, if you think the quality scores are important, maybe you don't like that. I mean, it apparently has worked out well for the Buckler lab to ignore quality scores like that.
So, this is Rajneesh. I have a question. Suppose we have the crop where the genome is not available, so we can do the test SNP from without any reference genome.
Yeah, you can use stacks without a reference genome, and you can import to polyRAD from there.
And in the polyRAD, suppose we have the BCF file, but in the BCF file we don't have the allele depth and any counting, and this only just SNP is there. So that's okay.
You won't be able to import to polyRAD if you don't have the read depth. polyRAD doesn't have anything to work with.
So we need read depth, or we can do without read depth?
You need read depth.
Okay, so it means we have in the BCF there should be the read depth of the each SNPs, yes?
Yeah.
Okay, thank you.
Lindsay, could you explain a little bit more about how population structure works when doing SNP calling? Does the method assume distinct allele frequencies for each subpopulation, and how does each
yeah, so the population structure method assumes it's more of an isolation by distance method, where it assumes there's sort of a continuum of allele frequencies across the population rather than discrete subpopulations. So it runs a PCA, and then it sort of determines how many PCA axes to use, and then it regresses the allele depths basically onto those PCA axes to estimate an allele frequency for every individual. For the time being, I'm going to keep going with the script, and then maybe I can answer more questions later. Okay, so this is HN over HE is a kind of a new statistic that I came up with, but this is sort of a heterozygosity-based quality statistic, both for samples within your data set and for loci. So it's looking at the allele frequencies and read depth and sort of seeing how well things match in terms of how heterozygous they look. Actually, before I do that, I'm going to show what did we get when we did an import. So we have this sort of summary. It tells you how many taxa and how many loci you get, how many total reads. We put in the ploidy as being tetraploid right here, and it's confirming down here that it's listed as being autotetraploid. And let's see. If you want information about loci, there's this loc table part, which will have the chromosome and position. Yeah, and now I'm remembering, I think this was something that I added relatively recently. It will have the reference allele listed at each locus if you put in the reference genome here. Okay. All right, so I'm going to run this heterozygosity-based statistic. And if I look at the output of that function, basically I get a matrix of numbers for samples and for individuals. And so here I'm going to get the total depth for each sample. And then here I'm going to get, use row means on this HH matrix to get that statistic for each sample. And then here I'm going to plot what that looks like. So in general, there shouldn't be too much of a relationship between read depth and the HN over HE statistic. But it's good to just confirm that. So this dashed line at 0.75 is showing the expected value of the statistic under Hardy-Weinberg in an autotetraploid. So this is explained a little bit more in the manual, but basically if it were diploid, the expectation would be it's 0.5. If it's tetraploid, the expectation is it's 0.75. If you had a hexaploid, it would be 5/6. So it's always ploidy minus 1 divided by the ploidy. And the fact that most of them are a little bit lower than that suggests there's some source of inbreeding, which in this case probably comes from the population structure because this is a highly outcrossing species. But what we see is, especially, is that there are some individuals with values much higher than that. And those are individuals that are, in this case, these are interspecies hybrids. So this is a good way to detect those. If you also say, like we had some individuals that were much higher ploidy than expected, those might also be higher like this, although they probably wouldn't go above 1 like these do. If they were much lower than expected, that could be it's a lower ploidy than expected, or it's a highly inbred individual. So these two commands here kind of show me which individuals are above 0.8 or below 0.4. So in this case, I can look at those and be like, oh yeah, those are some that I previously identified as interspecies hybrids. So this is of course going to depend on your data set. And I set these thresholds of 0.8 and 0.4 kind of arbitrarily by looking at the distribution here. You could always do something a little bit fancier to detect outliers and determine what threshold you'd want to remove samples by. So here I'm filtering samples. So yeah, okay, I didn't run this command yet. So I'm going to subset that matrix and then see what row names are left. So these row names remaining are the names of the samples that I want to keep. And so that's why I give those sample names then to the subset by taxon function. So then if I look at my data again, now I see I have 246 taxa instead of 270, and everything else is the same. And so now I'm going to look at HIND over HE by locus. So I'm going to run it again just because that now I've eliminated the samples that I think are problematic. All right, so I'll calculate it per locus, and I'm just going to get a histogram to see what I'm looking at. So this 0.6 about matches the median that I saw for samples as well, reflecting a little bit of inbreeding due to population structure. And so there's this kind of normal distribution, but it's overlapping with a couple other things. So way down here, we have some that are very low. So those will tend to be markers where there's a strong amplification bias, or maybe they're actually mitochondrial or chloroplast sequences. But otherwise, for some reason, they're just looking a lot less heterozygous than expected. So those are kind of suspicious. Over here, we have this kind of bump instead of the normal distribution kind of trailing off like you would expect. And what this represents over here are paralogous sequences. So cases where two different tags coming from two different regions of the genome accidentally both align to the same region. So these are also problematic markers that we want to get rid of. They aren't behaving like we normally expect. So I can actually estimate inbreeding from where this peak is. So if I'm saying my HIND over HE peak is at 0.6 and my ploidy is 4, I can plug those in and say my inbreeding coefficient is estimated at 0.2 for this. And what I'm going to use that for is I'm going to get the expected HIND over HE distribution from this function, saying like, okay, if I had a perfect data set of nice autotetraploid markers and its depth and sample size was the same as this data set, how would I actually expect this distribution to look? All right, so I run that and it basically resimulates a data set based on my data set. And it shows me that distribution, and it also says, okay, 95% of observations were between these two values. And so that's what I'm going to use as a cutoff. And because it was a random simulation, they're slightly different from what I had previously written down in the script. So I'm going to set those as my threshold. And I can say, okay, if I only kept markers between those values, what would I get? Yeah, in this case, that would keep 60% of markers. So that seems pretty stringent, but this is actually a highly duplicated genome. So in this case, I'm expecting to get a lot of these accidental paralogs. So it might be different for your study species. And then I can relook at that histogram and just draw lines there to say, okay, which portions of the loci am I keeping? So again, this is a little bit stringent. You might look at it and be like, nah, I'd relax that a little bit. I can see this distribution, maybe the cutoff's over here. So that it's arbitrary, but in this case, I'm going to do what this function output told me to do, and I'll keep those cutoffs. All right, so I'm going to put those into this vector called keep loci. So if I say do a preview of the first 50 of those are the names of loci that are going to be retained after filtering based on this statistic. So then I will filter by locus. And if I look at it again, all right, now it's reduced the number of loci. All right, and so now I'm going to do something called looking at the overdispersion of the data, which basically says, okay, now that I've filtered down to what I think are the nice quality markers, how well does the allelic read depth kind of look like what it's supposed to? Like if I have an ABBB individual, am I really getting a 1-to-3 ratio of reads, or am I getting something else? So basically that's going to be used for choosing an overdispersion parameter when we call the genotypes. So I'm going to test a series of different overdispersion parameters to pick the one that I want to keep. So it does a rough genotype estimation first, just so it can decide which are the genotypes that are going to test against those depth ratios. We might have time for like one question here while we, oh, never mind, it finished. I know when we do the genotyping, that will take a couple minutes. Okay, so then I can look at QQ plots of p-values generated by that function. And so what I'm going to look for is the plot where these black dots follow this red line for basically the longest amount. So if I set the overdispersion at 6, they peel off of it pretty quickly. If I go up to 8, they're a little slow to plot just because it's thousands of points. Yeah, now I see that those black dots come a little bit closer to that line. If I go to 10, right now it's even better. But if I go to 12, then it starts overshooting the line. Right, and this, it doesn't show up well because I have my screen so small, but on a bigger plot, you'd see that this is starting to sort of like loop over the line before going back down. So in this case, 10 is the one that I want to keep. All right, so here's the step where we actually call the genotypes. Right, so I've done my filtering, I've figured out how messy are my data, and I've put a number to that messiness. And so now I'm ready to go. And so this is iterative, like first it estimates allele frequencies, estimates genotypes from those allele frequencies, and then it estimates the allele frequencies again from the genotypes. And it does that for several rounds until the allele frequencies converge. So this is basically a small piece of the data set, so it takes a few minutes. If you have a large data set, you might, if you have access to a high-performing computer cluster, you might divide it up into several chunks that you can run in parallel. Especially, yeah, if you have like 1 million SNPs, you would definitely want to say break it up into chromosomes or something like that. If you have lots of time but only one little laptop, then you could break it into chunks that you run one after another. So on each iteration, it's telling me how many principal components did it use? How much did allele frequencies change from one round to the other? And by default, it will converge once allele frequencies change, I think by 0.0001 or less. Okay, so it's done running the genotype calling. So first I'm going to look at the population structure. And here I'm making a quick and dirty color scheme for my samples. So I'm going to have black as the default color. I know that if it's the samples that started with KMS came from Korea. So I'm going to make those red. Samples that started with JPN or JM came from Japan. So I'll make those blue. The ones that start with JY came from China. So I'll make those cyan color. And then if I run a plot function on the RAD data object itself and put in those colors as my colors, I get a PCA plot. Right, so I'm seeing that Korea and China kind of separated out on the first axis here. And the second axis then starts to split Japan into two different groups, which we know from experience, it divides itself into North and South. So I would want to take a quick look at that and make sure that it made sense and that it was something biologically real and not like a batch effect in the data set. Like, oh, it's dividing them into which PCR plate was it on or something like that. All right. And after that, so now I basically have the genotyping results in there, but they're maybe not in a format that would be easy to look at. So one thing I'm going to get is the posterior mean genotypes. And I use this get weighted mean genotypes function. And if I look at the structure of that, basically I have a matrix where rows are the samples and columns represent alleles. And so we have the chromosome and position of the allele and then the sequence for that allele. And by default, the most common allele is omitted for each locus. And that's because for various like statistics and matrix operations, if you included every allele, it would cause mathematical singularities that would be a problem. Right, and if you were doing a GWAS, you probably wouldn't care about the effect of the common allele, you're caring about effects of minor alleles. And I can preview what I get from that. And so in this case, it's numbers that range from 0 to 1. In the PowerPoint, I had them range from 0 to 4, just for ease of understanding. You could always multiply this by 4 if you wanted it to range from 0 to 4. Or I think there's also an argument you can change for that. So if I look at the distribution of those posterior mean genotype values, because I've omitted the common allele, most of them are 0, saying that there's 0 copies of the rare allele for that particular sample and locus. Then I have this bump around 0.25 for genotypes that have 1 copy out of 4. So it's like an AAAB genotype. That's what this is. And extends all the way to 1, but those homozygotes for the rare allele are very rare. I can get a preview of those posterior mean genotypes with a heat map. Well, that will take a minute to plot, but I don't recall it being too long. And I should say that this matrix, you could send it to a software like BGLR to do genomic selection with it if you wanted. Right, and so if I run this heat map, I see clustering of individuals and I see my North and South Japan and my Korea and China. So they're clustering in ways that make sense. And I can, by taking this bird's eye view of these colors, I can see how the genotypes are varying across the different populations. All right, and so I can also export to VCF. Now, this particular step is the one that will not work if you didn't have your reference genome file at the beginning. But it will work if you did have the reference genome file. So this takes a few minutes as well. I can do some questions while this one runs if there are more questions.
Yes, Lindsay, there's one here. Prior probabilities are not usually inferred from data in Bayesian models, as far as the question understands. So the question is, do you risk propagating errors by using the data to infer the prior probabilities from PCs on each individual?
I mean, I guess you do, but I don't know where else you would get the prior probabilities if you didn't get them from the sequencing data. Like, are you going to do a whole separate experiment to get allele frequencies at every locus across the genome before you run this experiment to do the genotyping? No one has the budget for that. I mean, and the way their prior probabilities is that the prior doesn't take into account the depth at that particular sample and locus, but the prior is looking at the whole rest of the data set, if that makes sense. Right, and if the PCA is derived from 10,000 markers and you're only looking at one marker, then the prior, I mean, it's slightly influenced by that marker, but mostly it's influenced by the rest of the data set. And the alternative is that you just have a flat prior and call genotypes directly from the read depth, but then at low read depth, that gets to be a problem because of undersampling of alleles. The lower the read depth is, the more the noise and the more those read depth ratios are going to deviate from the true allelic ratio in the genotype, the true dosage.
This is Rajnish. I have a question. With this polyRAD, we got, okay, the population has the 4 ploidy level or tetra or something. Can we get after this, separate the lines which is coming in which particular ploidy level, like diploid or tetraploid or triploid? Can we separate in this polyRAD or not?
In this case, if you have a mix of diploid and triploid and tetraploid, I would make those into separate RAD data objects and do the genotyping separately for each one. I mean, it's kind of been in my mind that, I want to ultimately have it be able to handle that, but I haven't implemented that yet. And I mean, the way it looks at the whole population, it would have to assume that all of these different ploidy levels are freely interbreeding with each other, which is unlikely to really be the case, even though there might be some gene flow.
Suppose we have the natural population for the 200 lines. Yes, in 200 lines, okay, we know these 15 lines is triploid and these 20 lines is tetraploid, and can be based on these known lines, we can predict the other lines something on this based on this something. Could you suggest on this? Okay, I'm sure these 20 lines is the tetraploid. So can I look the other lines in the rest 200 collection, which can be more tetraploids, same ploidy level and some triploid and something like that?
Yeah, right now polyRAD won't do that. It would kind of be each one in isolation. But I know what you mean, where you would want, especially with sample sizes like that, you would want to inform one from the other one. Yeah, maybe at some point I'll rework things to add that in, but right now it doesn't work that way.
Okay, thank you.
All right, okay, so this has finished running. And now if I look in my files, I see that I have one which is the genotype calls. So that has been exported to VCF. And I'm just going to open up my terminal. Oh yeah, I have my command where I ran it before, but and if you don't have this less command available to you in the terminal, don't worry about that for today. It's not essential for getting polyRAD to work, but it allows me to preview this file that I just created. So I'm hitting the space bar to scroll through it. So I have information about each sample recorded. And then I have the genotypes table. So here the samples are as the column names. And then here I'm starting to see the actual genotypes. Like, so here's an AABB genotype. It's listed as 0011. And then the read depth is exported again, as well as the total read depth. So in this case, the posterior mean genotypes and the posterior genotype probabilities are not exported. And that's because things got converted back from haplotypes to SNPs, and it wasn't really mathematically possible to back calculate that way. But I knew that people really wanted a VCF export. So that right now the VCF export just takes the most probable genotype for every sample in locus and exports that in a SNP-based VCF. Okay, I think, oh, actually, that's almost all I wanted to demonstrate. I did want to point out, so we're going to see some other software in the next couple days. And I wanted to point out that in addition to this exporting the VCF, there are export options for a lot of that other software, like MapPoly, PolyMapR, GWAS Poly. You can export directly to the file formats that those need. Okay, so yeah, now I've got time for more questions. Or
This presentation has been sponsored by the Tools for Genomics Assisted Breeding in Polyploids Research Project. For more information, you can email us at toolsforpolyploids@tamu.edu or visit our website at www.polyploids.org. This project is funded by NIFA within the USDA Specialty Crop Research Initiative funding program.